Hello everyone, welcome back to the class. In our previous class, we had learnt about transport of proteins into chloroplast. So the proteins which are to be transported to the chloroplast contains a particular sequence which is known as transit sequence, that is N-terminal signal sequence. That sequence is known as transit sequence, responsible for translocation of protein into the chloroplast. And there are two different complexes, translocone complexes. Two different translocation complexes, that is TOC and TIC, responsible for easy passing of proteins inside the chloroplast. TOC stands for translocone at the outer envelope membrane of chloroplast, and TIC stands for translocone at the inner envelope membrane of chloroplast. So the protein translocation starts by binding of transit sequence of protein to outer membrane receptor, that is TOC 159 and TOC 34. Both these receptors are members of family related to guanine triphosphatases with overlapping functions. So bound GTP favors this binding of transit sequence. So this forms the ternary complex. Ternary complex of the transit sequence with both TOC 159 and TOC 34. And this ternary complex is stimulated by GTP hydrolysis. GTP hydrolysis. The GTP hydrolysis opens the translocation pore or promote binding of TOC to TIC. So after GTP hydrolysis, it opens the pore or it binds the TOC and TIC complex to form a complete continuous pore so that the protein can get inside the stroma. After that, the pore seems narrower than those of the alpha helical seq translocone. The pore across the inner membrane consists of complex of at least 7 TIC proteins. The abundant protein TIC 110 not only forms some or all the pores but also binds to HSP 70 on the stromal side of the membrane. The ATP hydrolysis of HSP 70 in both the intermembranous space and stroma will promote the translocation of imported proteins straight to the stroma. So it will help protein to get inside the stroma. Now protein is inside the stroma, and remember, protein is having the transit sequence attached to it. So now after entering into the stroma, the signal peptidase will cleave off this transit sequence before proteins folds or redistribute to their final location. So there are other HSPs like HSP 60, HSP 100, which acts on different types of proteins and they remain on the stroma. Other proteins move on to thylakoid membrane or thylakoid lumen based on different pathways. The proteins which are to be translocated to thylakoid is having secondary N-terminal signal sequence. Secondary N-terminal signal sequence after the transit sequence is cleaved off. Some move across the thylakoid membrane into the thylakoid lumen through a translocone. After translocone, after the translocation, the peptidase in the thylakoid lumen removes both types of secondary signal sequence. So this is what we learnt yesterday. So this is the flow chart explaining about the translocation of proteins to different regions of the inside the chloroplast. So here, starting from transit sequence binding to TOC 159 with the help of GTP hydrolysis, and it helps in formation of continuous spore, and ATP hydrolysis with HSP70 helping that protein which is having transit sequence to enter into the tick complex. After entering into the stroma, the signal peptidase cleaves the transit signal, and HSP60 and 70 helps in proper folding. So today we will be learning about peroxisome. So it is another organelle discovered by Christian de Duve. It is a single membrane bound small organelle present in all eukaryotes. It produces and consumes hydrogen peroxide. So they contain oxidative enzymes like catalases and oxidases. So the major function of catalase or oxidase is, first let us know about peroxisomal oxidase. Peroxisomal oxidases, what it does, it transfers hydrogen atom, to molecular oxygen. What it does, this oxidase will transfer the hydrogen atom to molecular oxygen, and this transfer forms hydrogen peroxide. What does the peroxisomal oxidase does? It transfers the hydrogen atom to molecular oxygen and forms hydrogen peroxide. So there is another enzyme that is known as catalase. What it does, catalase uses this hydrogen peroxide. So it uses hydrogen peroxide for what? To oxidize variety of other substrates such as phenols, formic acid, formaldehyde and alcohol by peroxidation reaction. So this catalase use hydrogen peroxide to oxidize certain substrates, by the help of peroxidation reaction. So when there is excess of hydrogen peroxide accumulates in the cell, so this catalase converts this hydrogen peroxide to water. Okay, so the major function of catalase is to catalyze or convert the hydrogen peroxide to water if there is excess hydrogen peroxide accumulated in the cell. So the peroxisomes lack DNA and ribosome. All peroxisomal proteins like peroxisomal matrix and membrane proteins are encoded by nuclear genes synthesized on ribosome present in the cytosol and then incorporated into pre-existing peroxisome. In this, it is an organelle in the leaf cell. So this is stonoplast. Here it is mitochondria, it is vacuole, and here you can see peroxisome. Mitochondria is present, chloroplast is present. So all major important organelles are present in the leaf cell. The ability of peroxisomes to divide themselves suggests that these may have an endosymbiotic origin similar to mitochondria. So peroxisome undergo binary fission, so they divide themselves. So this suggests that it may have originated from endosymbiotic origin similar to the mitochondria and chloroplast. So the localization of peroxisomal proteins in the ER, which is similar, and the similarity of some peroxisomal proteins to those localized in the ER, suggests some alternative hypothesis that peroxisome has developed from ER, so de novo origin. So the peroxisome has two type of origin, we can say, according to this. One is symbiotic origin or de novo origin, developed from ER. So most peroxisomal membrane proteins are made in the cytosol by membrane bound ribosomes of ER and then integrated into the ER membrane, from where they may bud off in specialized peroxisomal precursor vesicle. New peroxisome precursor vesicles may then fuse with one another and begin importing additional peroxisomal proteins synthesized by membrane-free cytosolic ribosome to grow into mature peroxisomes, which can enter into a cycle of growth and fusion. So here this model explains how peroxisomes proliferate and how new peroxisome arise from endoplasmic reticulum. Here you can see the peroxisomal precursor vesicle bud off from the ER. At least two peroxisomal membrane proteins like PEX3 or PEX15 follow this route. The machinery that drives the budding reaction and that selects only peroxisomal proteins, so peroxisomal precursor proteins, that are still unknown. So here this is the precursor vesicle to which the peroxisomal precursor proteins will join within it, specific proteins that catalyze protein import. After that, the growth of precursor vesicle will happen by uptake of specific peroxisomal proteins and lipids from the cytosol. Then it somewhat divides into two, or with the help of fission it divides into two. So the major events or the function of peroxisome is the process of beta oxidation takes place, photorespiration and glyoxylate cycle happens in this peroxisome. So the same image showing different types of organelles present in the plant cell, lipid droplet, glyoxysome. So glyoxysome is a specialized form of peroxisome where a particular event in plant cells happen, that is glyoxylate cell. So this type of glyoxysome or specialized form of peroxisomes is majorly present in cells of germinating seeds. So they contain enzymes of glyoxylate cycle which help to convert stored lipid into carbohydrates that can be translocated throughout the young plant to provide energy for growth. After germinating seed, so the plant will develop, will proliferate, or will undergo this growth, right? So the energy is provided by this glyoxylate cycle by what? Converting the stored lipid into carbohydrates. In our next class, we will be learning about targeting of peroxisomal proteins from cytosol to peroxisome. Thank you.